All right. Thanks for the reminder, Nasima. And please feel free to jump in or interrupt me at any point while we're going through these notes. There is a lot to go through, and I'm going to be going pretty fast. So if there's anything that you want me to go over again or anything that you want me to clarify a little bit, please feel free to slow me down and let me know.
Okay, thank you, Mr. Michael.
No problem. Thanks for the reminder. So like I was saying, A and T, G, and C always line up exactly like that. So if you know what one side of the DNA looks like, you can use that to figure out what the other side is supposed to look like. A always goes with T, T always goes with A, C always goes with G, G always goes with C. When you're dealing with DNA and DNA, this will always be the case. And then we have those hydrogen bonds that hold the bases together. So I think this is a C and this is a G, maybe. The bonds themselves are pretty weak, but there's so many of them in a DNA molecule that they hold it together pretty strongly. So as we've talked about later on, the way that DNA works is that the series of bases act like a code, right? It's a code to make a certain protein. And so maybe if you're this alien and your code says CAT, then you end up with purple hair or purple skin or purple fur. On the other term, if yours says TAC, maybe that means that you'll end up with yellow hair. Maybe this is a different gene or a different type of gene that'll determine a different trait. So even though there's only 4 DNA bases, there's thousands and millions of different ways we can combine them together in order to create all of the different phenotypes, all of the different traits and characteristics that we see in animals and plants around the world. So after we talked about exactly what DNA is, the next thing that we went over was how do we make more of it? So oftentimes in the cell's life, as we're going to see later, it has to divide and make a new cell. That new cell needs the same type of DNA. And so we have to make sure that the new cell ends up with the exact same DNA as the old cell. So we have to copy the DNA, go through DNA replication, make a new set. So the way that DNA replication works is it's called semi-conservative, which is just a way of saying that each new set of DNA that we end up with is going to have one old strand from the original DNA and one new strand that we made in order to match up. So we have our original strand of DNA, it's going to split, and then each split strand is going to get a new side of DNA added on to the other side. So we call that semi-conservative replication. DNA replication happens in part of the cell cycle called S phase. So later on, we'll talk about mitosis and how exactly the cell splits into 2 or more. But we actually copy the DNA way earlier on in here in something called the S phase. So S stands for synthesis, which is just a fancy way of saying you're making something. So the S phase in the cell cycle means DNA synthesis. So the main way that we kind of kept track of what's going on in DNA replication was by thinking about the different enzymes that are involved. Enzymes are special proteins that do a certain job in a cell. And there were 4 enzymes that we talked about that do this important job of copying the DNA. The first one and kind of simplest one is called helicase. And all helicase does is take that double-stranded DNA and separate it apart. It's kind of like a little wedge traveling through the DNA and opening up those strands to go from this single double set of DNA to these two kind of individual strands with the bases out in the open. So helicase unwinds the chain and breaks those hydrogen bonds right here in the middle. However, once helicase goes through, we can't just immediately start copying DNA. One of the weird things about the DNA enzymes is that they need a starting point. They need something that they can start attaching their new bases to. So we create some little RNA primers to work as the starting points. And the enzyme that does this is called primase. So after the DNA gets opened up, primase comes in and puts down a short little RNA primer. That way, the other enzymes can attach and fill in the new DNA that's being made. So this RNA primer acts as like a starting point for the rest of the DNA replication to happen. And the primer is put down by another enzyme called primase. One of the tricky things about DNA is that it can only build bases in one direction. And it's called the 5' to 3' direction. You don't need to remember exactly what 5' and 3' mean. They're essentially opposite ends of the DNA. So if one end of the left DNA, the 5' end is at the top, that means the 3' end of the left DNA is going to be at the bottom. Also, if on the left strand, the 5' is at the top, that means on the right strand, the 3' is at the top. So the 5' and the 3' are always on different ends from each other. And DNA can only make new DNA in the 5' to 3' direction. So for one side of the DNA, called the leading strand, this is not a problem. So if we're looking at our example over here on the right-hand side, helicase is coming towards the right. It's going this way. And the 5' end is over here, and the 3' end is over here. So on our leading strand, we just need one short little primer, and then the rest of the DNA can come along and get filled in easy peasy. As more bases open up down here, more bases can be added, no problem. That's called the leading strand. The lagging strand is a little bit different. It's made up of a whole bunch of these little short segments. And on the lagging strand, it does this because the 5' and 3' are going the opposite direction from where we're making new bases. So we're constantly having to put down a primer and fill in some DNA, and then jump back a little bit and put down a primer and fill in some DNA. So on the lagging strand, we're going backwards, essentially. We keep traveling in the opposite direction. So we keep having to jump back and fill in these little spaces called Okazaki fragments. So polymerase 3 is the DNA polymerase that does most of the work here. So polymerase 3 puts down all of the DNA on the leading strand and all of those short fragments on the lagging strand. That's DNA polymerase 3 as well. So anytime we have some open bases and we need to put down new DNA, polymerase 3 is our guy. Eventually, we got all the DNA copied, but we still have those little RNA primers left over. We don't want that RNA in our DNA, so polymerase 1 comes along, cuts out those RNA primers, and fills them in with DNA. So essentially, polymerase 1 is responsible for cutting out these little green RNA primer sections and filling them back in with red DNA, the stuff that's supposed to be there. The helicase opens the DNA. Primase puts down those little primers. Polymerase 3 fills in all of the major gaps. And then polymerase 1 takes out the primers and puts in DNA. So again, we have our leading strand, which is going along smooth as silk. Polymerase 3 is putting down all that DNA. And then on our lagging strand, we're having the primer and then filling it in, and putting down a primer and filling it in. And then eventually, Polymerase 1 will come along, take out that primer, and put down some nice blue DNA. So I think that's pretty much all you need to know about DNA replication. All right, so that was kind of the first set of things that we started going over in class. Next week, we talked about, all right, we have this DNA, we know how to make more DNA, but what if we want to use the DNA to make those genes? What if we want to make something out of our DNA? Well, it happens through something called protein synthesis. So protein synthesis is the fancy name given to the overall process of taking the information in DNA, copying it to a strand of RNA, and then using that RNA to make proteins. So as we've just been saying, DNA is a whole bunch of these nucleotides all stacked up together. And certain sets of those nucleotides are called genes. So each gene is the code for a specific protein. So one gene makes one exact protein. Proteins are extremely important for our bodies. They help build cells, they help cells with their functions, they do a lot of the kind of behind the scenes inside work inside of our cells. So most of the cells and most of the jobs that are done in your body are done by proteins. So DNA has the information to make the proteins that we need. The way that we make proteins is that we take amino acids, which are a type of molecule. We link a whole bunch of them together and fold that all up, and it becomes a protein. So this protein synthesis is the process by which we take the information from the DNA, use that to figure out which amino acids that we need in which order, and then link all those amino acids and fold them up together in a protein. So our DNA is found in a portion of the cell called the nucleus. It's this extra protective structure that has the DNA inside. The tricky part is that the ribosomes, which make the proteins, they're like our protein factories, our ribosomes. They are not inside the nucleus. They're hanging out on the outside of the nucleus in a couple different places. They might be floating around on their own. They might be attached to another organelle, another part of the cell. But regardless, they are not where the DNA is. So we have the instructions in the nucleus, but the factory is on the outside of the nucleus. DNA cannot go out of the nucleus. That would be very bad. And ribosomes are too big to go inside the nucleus. So we've got to figure out some way to copy our DNA code, take that code out of the nucleus and to the ribosomes so that we can make proteins. So protein synthesis is this whole process, copying the DNA code, taking it to the ribosome, and crucially, making the specific protein that our gene is giving us the instructions for. So DNA is like our original, it's our master plan. RNA is like the copy, it's the blueprint. So we're going to use RNA in order to accomplish this goal, to copy the DNA and take that message to the ribosome. There are three major ways that we talked about that RNA and DNA are different from each other. They're both nucleic acids, they're both made out of nucleotides, but there are a couple of major differences. First one is the sugar. So every nucleotide is phosphate sugar base. In RNA, we have the ribose sugar, but in DNA, we have the deoxyribose sugar. They're a tiny bit different in structure. Ribose has this OH, DNA just has the H. But their sugar part of the nucleotide is different. Difference number one. DNA, as we've already talked about, has A, C, G, and T. RNA has A, C, G, and uracil, which we represent with a U. So 3 out of the 4 bases are the exact same, but there is no T's in RNA. Instead, we have U's. So whenever we're making the copies, the T in the DNA still matches up with the A, the G still matches with the C still matches with the G. But if DNA has an A, then that's going to match up with a U and the RNA. Again, RNAs do not have T's, they have U's. That's difference number 2. And then finally, difference number 3, DNA is a double-stranded molecule, whereas RNA is single-stranded. So here we have our DNA, which has that deoxyribose sugar and the T base. Here we have our single-stranded RNA, which has the U sugar, or has the U base and has that ribose sugar. So different sugars, different set of bases, and double-stranded DNA versus single-stranded RNA. So again, A and T and C and G always go together on DNA, but when you're matching that up with RNA, A will go with U instead. So there's 3 types of RNA. rRNA makes up ribosomes. We're not really going to talk about that one. We're mostly going to focus on mRNA and talk a little bit about tRNA. So mRNA is our super duper important one. It is what is responsible for copying that code from the DNA and taking it out of the nucleus to the ribosome. So it copies the DNA and then leaves out of the nucleus. And again, it copies that down exactly, except it swaps a U anywhere that a T should be. So we have our DNA here, and then another enzyme comes along, opens it up, and starts copying mRNA. So again, if the DNA has a G, RNA is going to have a C. DNA has a C, RNA is going to have a G. DNA has a T, RNA is going to have an A. DNA has an A, RNA is going to have a T. Nope, RNA is going to have a U. So the exact same setup as if we were copying DNA, but no T's in mRNA. Instead, we swap out anywhere that a T would go with a U. So after we make our mRNA, that then heads out of the nucleus and over to the ribosome. As the mRNA makes its way through the ribosome, through that protein factory, the ribosome is reading the mRNA and 3 bases at a time in something called a codon. So it's a 3-base code, sometimes called a triplet code. And each codon corresponds with 1 type of amino acid. So there's 20 different amino acids out there, and each one corresponds with a type of codon. So as the mRNA is being read, the ribosome is reading it 3 bases at a time, or one codon at a time. So the codons are important because they tell the RNA what base or what amino acid that we need next. But the thing that's actually responsible for attaching the amino acid to the protein is our tRNA. They have kind of a funky cloverleaf shape. The amino acid attaches at kind of the top part, and then down at the base part is something called an anticodon. So here is our funky shaped tRNA. Up at the top up here is where an amino acid would attach. And then down here is our anticodon. So this is going to match up with the codon and the mRNA. And essentially, it's going to confirm that we've brought the right amino acid in. So as the ribosome goes through codon by codon by codon, it reads the codon. In this case, it reads ACU. It then brings in the tRNA that has the correct amino acid. They match up with codon to anticodon. So ACU is going to match up with UGA. Because remember, A and U go together, and C and G go together in RNA. And then the TNA is going to drop off its amino acid, head out. The ribosome is going to hop down to the next codon, read that, bring in the next tRNA, so on and so forth until we reach the end. So the anticodon of the tRNA matches up with the codon our mRNA. If we want to know which codon will make which amino acid, scientists have created a whole bunch of different ways of seeing that. Sometimes you might see a chart like this, where you go from the big letter in the middle to the out to further out to figure out which amino acid is going to go next. Or more commonly, you'll see a chart that looks kind of like this, where you take your first position over here and your second codon or part of the codon up here and your third base over here. So for example, if we had AUG was our codon. A is the first letter. So we look over here on the left. Okay, here's our A row. U was our second letter. So, okay, U up here is in that first column. So we know that we're going to be looking somewhere in here. And then G, third position, is the one down at the bottom. And in this case, AUG gives us met or methionine. So just by reading the first letter, second letter, third letter of the codon, we can figure out exactly which amino acid that it's going to give us. So as the mRNA is traveling through the ribosome is reading each codon, bringing in the correct tRNA to match, and then that amino acid, whether it's methionine or alanine or threonine, gets added onto the chain, and then the tRNA leaves, and a new one is brought in for the next codon. And so we slowly build up this longer and longer and longer amino acid stretch until eventually we hit one of these codons, like UAA or UGA, that tells the ribosome, stop, this is the end. We can stop making the protein now. So again, we start off in our nucleus, where our DNA is copied down by the strand of RNA. So a specific gene for a protein is copied. That RNA then takes that gene out to a ribosome, and then the ribosome reads codon to codon to codon to codon, brings in the correct TNA, it drops off its amino acid, and we slowly grow this amino acid chain until finally we reach the end of the mRNA, and we can turn that chain into a protein. So DNA to RNA to protein. copying the code, reading that code, and putting the correct amino acid in the correct place to give us a protein. All right, so all of those steps are basically everything that we went over in week 3. And then in week 4, we started talking about meiosis and mitosis, the processes of cell division, where we also started to get into the idea of chromosomes in our cells. So this DNA that's hanging out in our nucleus, you don't just have one big, long DNA strand floating around in there. Instead, the DNA gets packaged up into these bundles called chromosomes. So chromosomes are the segments of DNA in our nucleus. They come in pairs called homologous chromosomes. So again, homologous means like the same copy or the same pair. And in humans and a lot of other organisms, we get one of our chromosomes from each parent. So the reason we have pairs of chromosomes is that you get one copy of all of your chromosomes from your mom and one copy of all of your chromosomes from your dad. So for chromosome number 1, you have a mom copy and a dad copy. For chromosome number 9, you have a mom copy and a dad copy. For chromosome number 17, you have a mom copy and a dad copy. They're the same chromosome. They might have slightly different types, slightly different forms of a gene on them, but you inherit each one from a different parent. And we call them homologous chromosomes. We've talked about genes already. So genes are sections of DNA that code for a certain protein, a certain trait. So each gene on your chromosome is a small segment of the chromosome that has the instructions for one protein. So before we divide our cells, first we have to copy our DNA, just like what we were talking about. And when we copy the DNA, now we have the exact same copy of a chromosome, and we call that a chromatid. So you start off with one chromosome from your mom and one from your dad, and then those become copies, and those copies are called sister chromatids. So 2 of the exact same chromosome where one has been directly copied from the other one. So the number of chromosomes that you have in your cells depends on which species that you are. So different animals and plants have different chromosome numbers. And it also depends on what type of cell that you're looking at. In humans, 99% of your cells are called somatic cells. They're like your normal body cells. And they have what's called the diploid number of chromosomes, meaning that they have the 2n right here, means that they have 2 copies of every chromosome. Just like we were looking at the pictures earlier, they have one mom and one dad copy for each chromosome. However, certain cells called gametes, which are your sex cells like sperm in males and eggs in females, they only contain half of the chromosome numbers. And because they have half, they're called haploid. So haploid means you only have one set of chromosomes. Diploid means that you have 2 sets of chromosomes. There are a whole bunch of different numbers of chromosomes in different species, but most importantly for us, humans, a normal human cell should have 46 total chromosomes inside of it. So humans have 23 different chromosomes. And so in 99% of your cells, you have 23 chromosomes from your mom and 23 from your dad. And that gives you a total of 46 chromosomes. So there's 23 different chromosomes in your cells. And in your haploid cells, you only have one single copy of each one. So haploid cells, those sperm and egg cells, only have the one copy, so they only have 23 total. 99% of your cells, your diploid cells, your somatic cells, they have 46 total chromosomes. So 23 pairs gives us 46 total. So the reason why we talk about these chromosomes and these chromosome numbers is that it's super important for cells to keep track of this when they're getting ready to divide and make more copies of themselves. So a lot of times cells will do this in order to grow or repair or replace dead or damaged cells. But in certain cases, we can also go through cellular division if we're getting ready to make those haploid sex cells that are going to come together eventually and make a new offspring. So if we look at the cell cycle in a little bit more detail again, the S phase we've already talked about. This is where we copy the DNA. Before that comes a G1 phase, which is like right after a cell is made. It goes into this, it's this normal growth period. Then after we copy the DNA, we go through the G2 phase. The other stuff in the cell is copied as well. And then we get ready to split the cell and pinch it apart. So the G1 phase and the G2 phase and the S phase are pretty long in terms of the percentage of time the cell goes through it. But then actually splitting the cell apart is a very fast process that cells go through very quickly. So there are two major types of cell division that we talked about: mitosis and meiosis. Mitosis is like your normal, everyday, average cell division. Whenever you just need to make a simple copy of your body cells, that is what you need mitosis for. So you start off with a cell with 2 copies of every chromosome. It replicates the DNA, it splits it apart, and then you end up with 2 cells with 2 copies of every chromosome. So they're identical to each other, they're identical to the original cell. You basically have just made a simple copy. That is what mitosis is all about. Again, I try to remember it is different from meiosis because mitosis has like 2 here in the middle, because in mitosis, we're making 2 cells. So when the cell is getting ready to actually physically split apart and go through mitosis, there's 4 stages that we go through. We have prophase, which is kind of like our getting ready phase. The chromosomes crunch up together and become thicker. The nucleus starts to disappear. The asters and the spindles, the things that are actually going to physically pull the DNA apart, start to appear as well. So prophase is kind of like our preparation phase. Metaphase is our middle phase because it's when all of the chromosomes line up right up and down the equator or the middle of the cell. So our spindle fibers are coming in and attaching to our chromosomes, and they're getting all perfectly lined up so that all of the different chromosome copies line up along that middle part of the cell. So like right here, everything is aligned and in the middle center area. Then we go through anaphase, the action phase of the cell, when the chromosomes get pulled apart to either side of the cell by those spindle fibers. So they start off right along the middle, and then the copies get pulled apart. So each one of the sister chromatids, which is now its own chromosome, moves to opposite sides of the cell. So anaphase pulls apart our DNA. And then telophase is kind of like the finishing up part of mitosis. The chromosomes loosen up again a little bit. The nucleus starts to show up around the chromosome. Those spindle fibers and those asters that pull the DNA apart, they kind of start to disappear. We don't need them anymore. And the cell is physically starting to separate into two different cells. So that happens in telophase. And then finally, cytokinesis is like the final little pinch, the final little cut that separates the cells apart. So that cell membrane finally pinches apart from each other, and we end up with two distinct new cells. So we start off with a diploid normal body cell, and then we end with two identical diploid body cells. So mitosis, at the end of the day, produces two cells. These cells are diploid. They have two copies of every chromosome, and they are identical daughter cells, meaning that they're the exact same as each other, and they're the exact same as our original cell up here. So this is what mitosis is all about. Simply taking one cell and copying it and splitting it into two new identical cells. Meiosis is a little bit different, a little bit more specialized. So meiosis only happens in our gametes. So in males, it's our sperm cells, and females, it's the egg cells. But these are the only places where meiosis happens. Nowhere else in the body goes through meiosis. So instead of making two cells, we make four. And instead of those cells all being identical and diploid, now they're haploid, and they're different from each other. So the reason why this is so important is because eventually the sperm cell and the egg cell are going to come back together to fertilize that egg. That's going to eventually grow up into a new baby or a new puppy or kitten or whatever it might be. So we need only 23 chromosomes from the mom and 23 from the dad, right? I mentioned earlier that in your normal diploid cells, you get one chromosome from your mom and one from your dad. So we need to make sure that these guys are haploid, only have the one copy, so that when they come together and join and fertilize together, we can make one single cell that is diploid, right? If both of these guys were diploid and had 46 chromosomes, when they come together, they make a cell with 92 chromosomes. And a 92 chromosome cell is not going to develop into a human. So in order to make sure that when fertilization happens, we have the right number of chromosomes, we need to take our diploid cells and split them into haploid cells. Only have one full set of chromosomes. So an egg cell and a sperm cell each have only 23 different chromosomes compared to the 46 total in a normal human cell and in this diploid fertilized zygote over here. So meiosis is pretty similar to mitosis, except it happens twice, essentially. So the DNA replicates and then splits apart, but then it splits apart again. One interesting thing that happens sometimes in meiosis is called crossing over. Essentially, it happens when you get all of these chromosomes that double up and are lined up right next to each other. Sometimes the DNA accidentally switches, and you end up with some of the DNA on one chromosome on a slightly different one. So because they're the same chromosomes, they just have a little bit different alleles or genes in them. Sometimes the DNA gets crossed and they swap pieces. And that only happens in meiosis. Mitosis doesn't have this happen. So meiosis 2 is just the second division where we have the DNA that got doubled and then separated, but then it gets separated once again. So we start off with a diploid cell, 2 big and 2 little chromosomes, and we split again into 4 haploid cells that each only have 1 big and 1 little chromosome. So we start off with a diploid cell with 46 total chromosomes, and now we have a haploid cell that only has 23 total chromosomes. And again, each one of these haploid cells is different from the other, right? None of them are the exact same, which is something that's different in meiosis versus mitosis. So again, a little bit clearer one here. We have, and again, 1 big and 1 little chromosome. So we start off with 4. They duplicate. There's some crossing over that happens. And then they divide once. And then they divide again. So we start off with 4 chromosomes. And now we only have 2. 1 big and 1 little chromosome. We end up with 4 cells in total that are haploid. And these 4 cells are all different from each other. None of them are the exact identical copies of the original. Independent assortment is another thing that we talked about. Essentially, it's just a fancy way of saying that the chromosomes don't split apart equally every time. So there can be some variation in terms of which chromosome ends up on which side of the cell and ends up in which haploid cell at the very end. So the mom's and dad's chromosomes don't always go to the same side of the cell. Instead, there can be a lot of variation in terms of which of these daughter cells get which combination of genes. So in summary, mitosis only goes through 1 division. Meiosis goes through 2. At the end of it, mitosis makes 2 identical diploid cells, whereas meiosis makes 4 unique haploid cells. And mitosis happens in order to have animals and plants grow and develop new cells and replace cells that die or get damaged. Whereas meiosis only happens in order to create new gametes, new sperm and egg cells. No other cells go through meiosis. Only sperm and egg cells go through it so that they can be haploid for the chance of fertilizing and making a diploid zygote in the future. All right, so there's some other stuff on these slides that you guys don't really need to know for the final. The main thing is just the major differences between the steps of mitosis and what happens in mitosis versus what happens in meiosis. All right, so that's most of what we talked about in week 4. And then in week 5 is when we started talking about genetics and how exactly it is that those genes come together in different ways in order to create different types of traits. So there are a couple different things that we talked about when we talked about genetics. The first one was the 2 different ways that we can describe a trait that you get from your genes. So you can describe the phenotype, which is the physical expression of the gene. So phenotype because it's physical. So what somebody looks like, what their hair color is, what their eye color is, if they have freckles, if they have dimples, or if they have a hitchhiker's thumb. These are the phenotypes. These are things that you can see or observe about a person. You can see their traits. The other way that you can describe a trait is through the genotype. So the genotype is the 2 letters like big B, little b, or little r, or big F, big F. Those are genotypes. And a genotype represents the allele pair of that trait. So alleles all are alternate forms of a gene. So like we were saying earlier, one of your chromosomes and sets of genes you get from your mom, and one set of genes you get from the dad. And they don't always have the exact same trait in their gene, right? Maybe your mom had the gene for blonde hair, but your dad had the gene for brown hair. So there's different versions of a gene. We call them alleles, and we represent them with big or little letters, depending on their dominance. So even though those homologous chromosomes that you get from your mom and dad both have the same genes, they might both have the hair color gene and the hair type gene and the eye color gene, but those genes might be in different alleles, different forms. So phenotype, physical expression that you can see of a gene. Genotype is the actual alleles that are interacting to give you that gene. So, for example, if we're thinking about freckles, your phenotypes are either you have freckles or you do not have freckles. So again, if you don't know, freckles are like the little pale dots that people sometimes get on their skin. So either you have them or you don't. Those are our phenotypes. A genotype would consist of, in this case, 2 letters F. And having freckles is the dominant trait, and not having freckles is the recessive trait. What that means is with the dominant allele, the dominant trait, that means as long as you have that allele even once, you will get the trait. So anybody that has at least one big F, no matter what the other allele is, big F, little f, they are going to have freckles. Not having freckles is the recessive trait. And the recessive trait only shows up if there's no dominant alleles that appear. So for somebody to have no freckles, they would have to have a little f for their trait. So if you're big F, or big little f, you have freckles because big F represents a dominant trait, which in this case is freckles. Only people with the recessive trait, 2 little f's, they are the only ones that will not have freckles. So one dominant allele is enough for the trait to show up, but if you want the recessive trait to show up, you have to have 2 recessive alleles. There cannot be any dominants that are there. Same thing like we talked about for hair color. So if brown is dominant and red is recessive, then if you get big R, or big R, little r, you're going to have brown hair because you have the at least one dominant allele. But if you have the 2 recessive, little r, you get red hair. So genotype is this allele pair, the letters. Phenotype is the appearance, what you can actually see about the person. So the nice thing about these genotypes is that they allow us to make something called Punnett Squares, which are ways where we can predict what is the probability that one of their children will get a certain trait. So for the first example that we did, we said that the parents are homozygous. So homozygous is a fancy term. That means that both of the alleles are the same. Like in our red hair example from back here, big R, and little r are our homozygous genotypes because we have 2 of the same allele, 2 dominant alleles or 2 recessive alleles. So homozygous means that you have 2 of the same allele. So the mom is freckles homozygous, and the dad is no freckles homozygous. So the mom is big F, dad is little f. In order to make a Punnett square, you put one parent's genotype across the top and one parent's genotype down the side. In this case, we'll put the mom's across the top and the dad's down the side. And then you go through and fill in each box based on which alleles that are connected to it. So for example, this box is connected to a big F up here and a little f over here. This box is connected to this big F up here and this little f over here. And then same thing for this box and this box. There's big and little to make this one. And then this one comes from big and little. So no matter which way you rearrange their genes, big F, little f is the only possible outcome for these parents and their kid. That means that there's a 100% chance that their kids are going to have freckles. Even though the dad is freckle-free, all of their kids will 100% have freckles because they will all inherit at least one big F from their mom. On the flip side, if both mom and dad are heterozygous, heterozygous means the two alleles in the gene are different, meaning you don't have too big, you don't have too little. Heterozygous says you have one big and one little. So in this case, the mom and the dad are both heterozygous, so they're both big F, little f. So we set up the Punnett square the same way, one parent across the top, one parent down the side. And we fill it in the same way too. This would be big F, this one's big F, little f, this one's big F, little f, and this one is little f, little f. So now, if we want to know about their percent chance of having freckles, 3 out of the 4 boxes are going to give a child with freckles, right? All of these have at least one big F, so 3 out of these 4 will be freckles. 1 out of the 4 is recessive and will not have freckles. So 3 out of 4 is the same as 75%, and 1 out of 4 is the same as 25%. Great question, Elizabeth. She's asking if the mom always goes on the top or side. It doesn't matter. The mom or the dad could go up here. The mom or the dad could go up here. It might change around which letters go in which box, but the probabilities will end up being the same. So it doesn't matter how you set up the Punnett square in terms of which parent goes where. Great question. So again, 3 out of 4 chance that they get a dominant allele and get freckles, which is 75%, and 1 out of 4 chance that they get 2 recessive alleles and don't have freckles, and 1 out of 4 is 25%. Then we went through some more examples of putting together Punnett squares based on some information that we have, and some other traits that are based around simple genetics like this as well. But the main things to remember are phenotype versus genotype, what you can see versus the actual 2-letter allele pair, homozygous versus heterozygous genotypes. So homozygous means the same, too big or too little. Heterozygous means different, one big, one little. And then finally, the other major thing to remember is how to set up a Punnett square. One parent across the top, one parent down the side, and then you just fill in the boxes based on which alleles that are connected to that box. All right, the other thing that we talked about having to do with genetics was how to set up pedigrees. So pedigrees work kind of like family trees in order to show which family member got a certain trait. So usually the pedigree works by having males are represented by squares, females are represented by circles, marriages are represented by horizontal lines, and then their children in the next generation are the vertical lines. And then the people that have a trait are usually filled in on the box. The people who do not have a trait are usually left blank. And if somebody is a carrier, meaning that they do not have the trait, but they have one of the genes for the trait, they're sometimes shown with a half filled in box. So a female and a male are connected by marriage, and then they have some children. Filled in means that they have the trait, and half filled in means that they could be a carrier. We can then label pedigrees with some simple labeling. And so usually the generations are indicated with Roman numerals, and then the individuals within a generation are numbered with our standard numbering system. And then we represent people with a combination of their generation and their number. So for example, individual 1-2 is this guy up here. 2-4, we have 2-4 is right here. 3-5, generation 3, person 5. And 4-1, generation 4, person 1. So it's an easy way of pointing out exactly which person that you are talking about. We could have dominant pedigrees, which shows how dominant traits, big letter traits, are passed down. Usually in dominant pedigrees, a lot of people will get the trait because it's easy for it to show up in a person. Recessive pedigrees, again, a recessive trait only shows up when you get 2 recessive alleles. So this is where you can get carriers, people that have the dominant allele, so they do not have the recessive trait, but they still carry a copy of the recessive allele, so they could pass it down to their future offspring. So for example, both the mom and the dad here are big A, little a. So they do not have this trait, but because they're both carriers, they end up having a son that does have the trait. So even though they don't have it, the son does, because they are both carriers of that little a allele. Because of the big A, they don't have the recessive trait, but they're passed down that recessive trait to their son. So all of the traits that we've been talking about so far with the big F, little f, big R, little r, are on autosomal chromosomes, which are just the 1 through 22 normal chromosomes. Sometimes, though, genes can be sex-linked, which means that they show up on the X chromosome. So males have 1 X and 1 Y. Females have 2 X's, at least in humans. And the Y chromosomes don't usually have traits on them. So these sex-linked traits only show up on the X chromosome. So that means that while for females, the rule still works the same. They have to get 1 or 2 big letters to get the dominant trait and 2 little letters to get the recessive trait. Males, the Y chromosome doesn't count. So if they inherit 1 copy of the dominant trait, they're getting the dominant trait. If they inherit 1 copy of the recessive allele, they get the recessive trait. Males don't have a second allele on the Y chromosome there. So we looked at some different examples of sex-linked traits that you don't really need to remember about. The main thing to remember with the sex-linked ones, again, is just that the females that have the 2 X chromosomes, they still inherit the traits normally with the 2 different alleles. But the male children don't get any of those alleles from their dad. Since the male children only get the Y chromosome from their dad, it depends on if they get the recessive trait or the dominant trait from their mom. That's the person who will pass it on to the male children. All right, so that was how to read a pedigree. And then in week 6, we talked about evolution and cladograms. So with evolution, we talked about a couple different things, namely evidence from fossils and evidence from homologous structures. So evolution, or descent with modification or change over time, is this super gradual process that takes thousands of generations and millions of years, but it causes organisms to change from a certain type, a certain species, into a new species. So modern organisms are all descended due to these changes from these ancient organisms. So one of the things with the fossil record that we talked about that supports evolution is that there are things that existed in the past that no longer exist today, and things alive today that did not exist in the past. So the stuff in the past and the stuff in the present are different types of organisms. However, they're not completely 100% different. Even these extinct ancient millions of years ago fossils still resemble modern animals, modern plants and animals. So it shows this common ancestry, this idea that the animals from millions of years ago slowly changed and developed over time into the animals and plants that we have today. Also, we found that the further down you go in the rock, the older and more simpler fossils that you get. So all the fossils at a certain layer of a rock will look kind of similar. And then as you move up, as you move more towards the modern organisms, you can figure out what is the order in which these species developed and kind of show a timeline of first we have this, then it evolved into this, then that evolved into this, and then that evolved into the animal that we see today. We can also compare different structures between organisms to look at evolution. And the ones that do show a common ancestry are called homologous structures. They're different in function in terms of what the different animals and plants use them for, but they're built in a similar way. So the big example that we talked about was the leg bones in vertebrate animals. So lizards and birds and mammals all have the same general structure of their leg bones. One bone at the top that connects to two bones that connects to lots of little bones. Whether you're a human or a sheep or a dolphin or a bird, you find this similar set of structures in the limbs of these organisms. So because these are so similar to each other, even though they're used for different things, it suggests that at some point, millions and millions of years ago, there was some sort of four-legged creature that had this one bone, two bone, lots of bone structures. And then over time, that became different in the different animals, but it still kept that same basic outline, that same basic structure of one bone, two bones, lots of bones. So even though they're used for different things, they all look like they came from some common ancestor. On the other side, you have analogous structures, which are organs and structures that animals used in a similar way, but came from completely different evolutionary paths. So like the wings in insects and the wings in bats and birds, both used for flying, but they're built completely differently. Insects do not have any sort of bones or any sort of structure inside of their wings, compared to bats and birds, which do. So even though they're used for similar things, they are built completely differently, meaning that they don't show any sort of common similar ancestor. We also talked about vestigial structures, which are the structures that aren't really used in the organism today, but were important for their ancestors. So things like the wings on ostriches and penguins and stuff like that. The appendix in humans. The tiny leg bones in whales. Our ancestors had these and made a lot of use out of them, but the organisms that survived and evolved today don't really need them. So if we want to show how structures have evolved over time and how we can group animals and plants based on their structures, we can use something called a cladogram. So a cladogram is a type of diagram that shows these evolutionary relationships between organisms. The closer that organisms are on a cladogram, the more similar that they are and the more closely related that they are in terms of evolution, meaning that they more recently diverged from a common ancestor. So for example, humans and sharks have a common ancestor from hundreds and hundreds of millions of years ago. But humans and chimpanzees probably have a common ancestor from only 2 or 3 million years ago. So by sort of organizing out these different animals and or plants based on traits that they share, we can see who is more closely related to who. So cladograms are set up by having sort of the more ancient relationships closer to the bottom of the cladogram, and then you get to more modern common ancestors as you get closer to the top. And then each branch on the cladogram represents a different species. So where the different branches converge is where they share common ancestors. And then usually the one that is least similar to the other groups, or the one that's kind of the first one to branch off, is called the outgroup. So for example, here in this orange box, we have the common ancestor of A, B, and C, but then one group split off to become eventually A, another group split off to become eventually the common ancestor of B and C, which then split into 2 different species. So B and C are more closely related to each other than they are to species A. So again, by looking at where the species have a common ancestor and how closely to the present, so the further you get to the top of the cladogram, the further you get to the present. Also, sometimes, like you saw in the homework, the cladograms are flipped and they're turned on their side, but the same rules still apply. The closer that you are to the far side, the closer that you are to the present. So leopards and turtles have a relatively recent common ancestor compared to, say, turtles and lampreys from way, way back in the day down here. All right, we also made our own cladograms, but there aren't any questions on the exam where you have to make a cladogram, so we're going to skip over that part for now. All right, so finally, the last things that we talked about, I'll go ahead and say that there are not any camouflage and mimicry questions on the exam, since that was just kind of a fun thing that we went over at the very end of class here. But we did talk about natural selection and classification. So natural selection is essentially kind of the process by which evolution happens. So we were just talking about how evolution, you have this change in species over time, where you go from the more ancient species to the more modern one. Natural selection describes why and how these species are changing. So the idea is that organisms aren't all going to survive the same, and that only the organisms with the best traits in order to survive and reproduce are going to have lots of babies and pass down their traits to the next generation. So eventually, over millions of years, you could have a population change in its traits and in its characteristics based on whichever ones work the best in the environment in which they're living. We talked about 4 things that are needed in order for natural selection to occur. The 4 tenets is what we called them. And that is, you have to have different individuals in a population, so not everybody can be an exact clone of each other. Those differences have to be able to be passed from the parents to the offspring, so it has to be something that can be inherited. More organisms are born than can survive, so there's going to be some sort of struggle for survival, and not everybody is going to make it. But the ones that do make it are going to have the better adaptations and have more offspring in the next generation. And so eventually, you end up with the more that the best organisms survive and reproduce, their traits are the ones that get passed down. And eventually, over millions of years, you could entirely change what a species looks like based on the traits that are the best for their environment. And that's kind of the main principle of natural selection. We talked about the example of the peppered moth, where they started off as mostly gray, but then as the environment got blacker and blacker from all the soot and the dirt, the black moths started to become more prevalent because they started to be better at hiding from predators. We also talked about classification and how organisms are grouped into species and different groups. The main kind of takeaways from it are that there's a reason why we give animals and plants specific names, and that is to cut down on the confusion, because while their common name might be different in all languages, their scientific name is always going to be the same. So this way, it helps make it very, very clear exactly what animal and exactly what species that you are talking about. Called binomial nomenclature, which is just a fancy way of saying that animals and plants all have 2 names. So for example, we might call this a raccoon, but scientists around the world can identify it as Procyon lotor. So all animals and plants have a specific 2-name scientific name. And then we also talked about the different kind of classifications from super, super broad, like kingdom and phylum, all the way down to very specific, like family and genus. And then finally, we talked about dichotomous keys and ways that we can identify organisms based on their traits and kind of figure out exactly what species we're working with by following a key and using that to distinguish between similarly looking organisms. And that is everything that we talked about in Biology B this term that you may need to know on the final exam.